Now let's take a look at summarizing some of the things that happen with mitochondrial dysfunction. What's going on in our mitochondria? We know that they are especially prone to DNA damage, and there's even a whole theory, the mitochondrial free radical theory, that suggests that the more we consume and eat, the more cell respiration that goes on, and thus the more oxygen is split in order to form H2O, right, which generates, when we split the oxygen, free radicals that can go around and do damage, and the more of that damage that's done, the more aged cells become. Now, there have been some challenges to this free radical theory, or expounding upon that free radical theory. It really depends on how you look at it. But this is how I would summarize that information. So it turns out that not only do we have the idea that, yes, indeed, mitochondria do accumulate free radical damage throughout their lifetime, and that becomes more with aging. But is it purely because of the increase in free radicals or oxidative reactive oxygen species? So the question is, do these species actually cause our deterioration, or what else might be going on? So more recent evidence suggests that perhaps the mitochondria, in response to stress, right, if they're not functioning that well, or if there's not enough energy around, we would upregulate the production of mitochondria such that there are many more mitochondria. And as a consequence of that, the more mitochondria are producing more reactive oxygen species, or 1/2 O2s, that are able to go around and do more damage. But we are also increasing the energy output, so that might counteract that. Anyway, so we are as a congruent thing, we are growing more mitochondria in each cell, and they are also producing more reactive oxygen species. And so perhaps the increase in reactive oxygen species, or ROS, is due to the fact that there are simply more mitochondria. Either way, the reactive oxygen species are causing damage and are certainly aligned with cellular aging. So just a little note on some of the newer research, but free radical theory is sort of the leading idea at the moment. Moving on to the next physiological trait of aging, you are probably very familiar with the idea of telomere attrition. Certainly, we have covered that telomeres shorten as cells go through divisions, and stem cells and cancer cells might have telomerase active, and so they can continue to grow or regenerate telomeres. However, most somatic cells lose that capacity, and you're probably familiar with the Hayflick limit. Hayflick limit suggests that there are a limited number of cell divisions that somatic cells can go through before the telomeres become short enough to start doing damage to the, subtelomeric regions of the chromosome. Once the telomeres have essentially run out. It's suggested that 50 divisions is about the maximum for any somatic cell. So after that, each cell division is shortening the ends of the chromosomes till we start actually nibbling away at the genes located on the ends of the chromosomes, which, as you can imagine, can have some manifestations in the condition of the cell. So telomeric attrition definitely associated with the aging cell.